Welcome back. Well, there have been reports of a new type or a new variant of COVID-19 in the UK, with scientists saying that this new type of the virus appears to be able to infect cells in a different way to the main variant. At the same time, you will have seen the increasing number of COVID-19 infections that we saw over 10,000 new cases being reported overnight. To talk more on this, I'm joined now by Professor Adrian Purin. He is the acting head of the National Institute for Communicable Diseases. Professor, good afternoon to you. First, let's just talk about the numbers. Just a week or two ago, we were talking about 3,000 cases. Now we're talking about 10,000 new cases. It seems to me to have jumped really, really quickly.
Yes, we're on that exponential trajectory that we've seen in the first surge or first wave, if you like. And in fact, we're about, I think if I get the calculations correct, about 60% of that first peak, if you like. So yes, we are on that marked increase, especially in the 4 provinces, specifically the Eastern Cape, the Western Cape, and KwaZulu-Natal, and I think Gauteng is about to join that second wave as well. I think there are flickers in the other provinces, but those are the major provinces. So if you look at the heat maps, you're quite right. So if you look at the heat maps along the last 2 weeks, you can definitely see that shift from green to red, especially along the coast. It's quite remarkable to see the changes that we are seeing currently.
Do we know why it's changed so quickly? I mean, why it seems to be jumping? I mean, it seems certainly to me to be quicker than the first time around. And I realise our memories of sort of the hard lockdown change over time. But is it moving more quickly?
It appears to. In fact, I was just looking at some other papers. In fact, if you look at the waves in other countries, in fact, those can be fairly larger or more substantial. We've seen that in the United States as a good example. And I know that there are difficulties around the United States, but certainly if you look at the UK and some of the other countries, a very dramatic increase. So if you look at the South African situation, and if you look at the strict lockdowns and the risk-adjusted steps that have been taken, as we approach level 2 and partly level 1, the so-called effective R, the number of people that are infected by a single individual that is infected, was reduced to less than 1. But as the restrictions have been removed, so has the fact that our compliance or adherence to the so-called physical distancing, which is so critical, the wearing of masks even more so, hand hygiene, these factors have probably contributed, I think, largely to the fact that they have not been adhered to. And therefore, we're now seeing that we're above an effective R of 1. And that's seen in all those particular provinces.
Our understanding of the virus is changing so quickly, and I understand why. I mean, we're obviously learning more and more. There's more and more scientific resources being applied to it. There was a time when we thought, well, if we get through winter, we'll be okay in summer. And then we started to see the numbers going up in the Northern Hemisphere, the United States and Europe. And we thought to ourselves, well, look, that's proof. They're going through winter, and that's because people spend more time indoors. And so you see a lot more of transmission of the virus. Now we're seeing in our summer, numbers going up very quickly.
Well, Stephen, if you recall that even in the Northern Hemisphere, even during summer, there was continued transmission as well. The curves may well have looked flatter, just like our curve looked flatter for the last few weeks. But as things were also removed, those restraints were removed, we certainly saw those changes during their summer as well. And that's currently what we're seeing as well in South Africa. So this is a virus that's not the flu. It's very different from the flu in that sense that, flu, in fact, we've not even had a flu season this year, but the flu essentially disappears in summer, you're quite right. But this virus has been transmitting throughout our summer. And as we can certainly see now, we're heading towards that second wave, even before the winter has hit us.
This new variant that has been identified, and I don't know how important this is in the UK. I mean, is it very different from the variant, what I suppose we would call the main COVID-19 virus that we see here?
Yeah, my sense is that, this is a virus, it's an RNA virus, and RNA viruses very often have high rates of mutations, but this is a virus that actually has proofreading ability. So its mutation rates are somewhat slower and more stable. And we've certainly seen some dominant variants coming through, but there's no real evidence from the earliest stages, at least, that these variants are really critical with regard to what we call pathogenicity. In other words, it's a fitter virus and therefore it transmits and therefore causes more severe disease. I think those types of evidences have not always been available. And similarly, with all the current ways in which these viruses are being analyzed, and again, it's an incredible feat of what is being done, are the large numbers of viruses that are being sequenced and variations sought for, and looking at critical regions. Now, I'm sure, again, when you think about the vaccine, you know that it's the spike protein. And so that, again, they're looking at those types of changes within, for example, the area that the virus binds onto cells, and looking at the variants. Again, there are changes or mutations, and there may well be new lineages, but in NSC, we still need the evidence that these changes are really critical, that the viruses therefore become more fit and adapted and will cause more severe disease. And I think those are the types of experiments or evidences that we need to really say that we're now seeing a more fitter and more transmissible virus.
As I understand it, a virus can mutate. I mean, viruses mutate and they die and are reborn so quickly that they can mutate far more quickly than, humans evolve, for example. So would I be correct that a virus can mutate and become more aggressive and more dangerous, can also mutate the other way and become less harmful to humans?
Absolutely. So if you think about some of the polio vaccines are a good example of that. These are viruses that are actually attenuated and less fit, and therefore they are usable. But you're right that there's the opposite direction, that in fact the virus may adapt through time in terms of being exposed for longer in human transmission chains, that they actually become different and therefore difficult to control. So HIV is the sine qua non of a virus that has been with us for several decades now, and actually has evolved so rapidly throughout. In fact, we're still struggling actually to produce a vaccine, an effective vaccine.
You mentioned the phrase proofreading. Now, I'm a journalist. I know what proofreading means to me. What does it mean to a virologist?
So you're quite right. So if you had a sentence starting with A, B, C, right up to Z, what they're doing is to put together all the other viruses and sequence them. In other words, they're looking at the nucleotide sequence of those viruses, and it is in a specific sequence, and it codes for specific genes very often, or they may be what are called non-coding, but they may well be critical for control. So looking at those sequences, we would look at the first virus that was from Wuhan in China, and then compare all those particular sequences against that particular sequence, and look at what the changes are, and calculate how distant they are from that initial virus. And that creates what we call clades to look at how that virus has changed over time. So it comes back actually to your point. If we don't have the physical distancing, the wearing of masks, transmission is ongoing, and the virus certainly adapts, and that may have implications, for example, vaccine development.
Okay, so if I've understood, to use another example, your organization was very involved in tracking the listeriosis outbreak several years ago. I think 200 people died from that. It was because you could sequence the DNA that you could tell where it had all come from, and it had come from a particular meat plant in Polokwane. So you can tell if I contract the virus, you can sequence the virus, and you can tell that it comes originally from Wuhan because of that proofreading ability.
Absolutely. So again, if there are outbreaks, for example, we're looking for clusters. So are people who've been infected at the rage or linked to each other? If we could sequence all of those, we would definitely see that type of arrangement versus, for example, an event in Johannesburg. So if we have larger outbreaks, more people affected, are those individuals, do they share that particular sequence to what has happened in the rage in, say, KwaZulu-Natal? And therefore you create the links. So the epidemiological and molecular links are really critical to tell you how that virus has moved from one place, one individual to another, and look at the geographical distribution as well. So that's really key and really an important tool that we make use of.
So, following on from the headlines, and this new variant was found in Britain, so you can imagine the British newspapers and the meal they would make of a new variant. But it does lead to two other important questions. The first is, I would like to presume, please tell me, Professor, so that I can sleep tonight, that the test that we have for COVID-19 would pick up the new variant, A, and B, the vaccines that are being rolled out in some parts of the world would work against the new variant.
So as far as I'm aware, I haven't seen all the details, but coming back to your point about vaccines, as far as I understand it, this should not affect vaccine efficacy. But again, I think we need to keep that in mind, because that's the whole point, is that if there are key changes in terms of the binding and the vaccine is not directed against that, because it has now changed or altered, then that certainly becomes a problem. With regard to diagnosis, again, what one tries to do very often is to look for what are, if you like, stable regions within the genome, as it's called. And those are the areas where they're really critical, and changes in that area will cause the virus to be less fit. So we really target those particular areas or genes in terms of amplification. And very often, when I mean amplification, is the molecular amplification of that particular virus. And so those should be very stable, and that should not affect our diagnostic capabilities at all. And we know, and HIV is a good example of that. When they set out to diagnose HIV, they focused on clade B and specific regions, and therefore missed the other viruses. But now it's so designed, and the same applies to all these particular diagnostics, is that it's so designed that you're looking for stable regions that will not alter and mutate rapidly, so that you can always consistently amplify, and that it does not become a problem.
Professor,
you can sleep well tonight.
Thank goodness, as well as I'm going to. Professor, I do need to just ask, I don't want to get you into trouble, but the burning issue of the moment is, should beaches be open or not? I know Professor Salim Abdul Karim, for example, your colleague, has suggested that he somersaults on the issue because it's outside, but it's mass gatherings. Do you have a view?
Well, I may not have a specific view. I know that the provinces have to develop their nuanced approaches to provinces and how they manage that. And so I suppose if the concern is around, again, I'm probably flip-flopping on this, to use a pun, is that when you're looking at beaches, it's on the outside. But if people are in close contact and not in their safe bubbles, do the risks increase? And therefore, is that a rational decision around closing or limiting people's access to beaches? So I think it really comes around at an individual level. And I think the NICD has just recently, with its colleagues in the modeling consortium, released an app where you can look at your risks. So if I'm in the Eastern Cape, I can see which area I'm going to go to, what are my risks in terms of how the virus is transmitting in that particular region. And therefore, do I really want to go to the beach? Do I really want to travel? Do I really want to go to places X and Y where I know my risks are going to be increased? So I think it also comes down to markedly an individual level to understand risk and to manage that particular risk. I think we need to talk about safety bubbles. I think that to me is the key in a way. I think we don't talk much about it, but I think that it's really important that we say, well, I'm in my close-knit bubble. Do I want to merge with another bubble where I don't know what my risks are?
May I ask you for the name of the app? I wasn't aware that there was one already. Do you have it to hand?
I don't have it to hand, but I can share that with you.
All right.
And you certainly can make that available to yourselves and to the public.
Professor, thank you very much indeed. Professor Adrian Piran, I really do appreciate the time. The Acting CEO of the National Institute for Communicable Disease as well. You learn more and more about the virus as we go.